Okay, so my name is Bolger. I'm also the president of Math Society, so if you have Math Society related questions, you can come talk to me. I'm presenting on my research that I did this summer. The project is called the genomic prediction of traits using convolutional neural networks. This research was funded by the NSF, and it was a part of a research experience for undergrads, or an REU. It was at Michigan State University. Specifically, it was in Dr. Shin-Han's lab, and I worked closely with Christina Zodi. So just a brief overview. I'm going to talk about what is genomic prediction. I'm going to go over the format of the data. We're going to talk about past work, or the motivation for this project. I'm going to describe to you what a convolutional neural network is, and then we're also going to talk about how these CNNs fit into all the research that my group had done previously. Okay, so your first question should be, what the heck is genomic prediction? So genomic prediction is just taking the genetic information of a plant and getting it and predicted trait value from it. There's a lot of ways that you can do that. There's a lot of prediction models you can build. So you can build linear, nonlinear, machine learning. But as long as you have a model that takes in the genetic information and outputs a trait value, you're good to go. The reason we like to do this is because it saves plant and animal breeders a lot of time and money. So plant and animal breeders are typically looking for some optimal trait. So let's say they're trying to optimize the height of a plant. So having this program before they actually do the breeding process quickens up all the work that they have to do. So the next thing I want to talk about is how we actually get the data that we're going to analyze. So if you think back to the last time you took biology, maybe it was in high school, maybe it was when you were in college, but within each of us we have cells, and within those cells there's DNA information, and that DNA information contains ideas or things about traits about us, right? It's the same thing in plants. So this genetic information describes our eye color, our hair color, how tall we are, things like that. If we have two varieties of the same species in a plant, their DNA structures will be most the same, meaning they'll look the same for the most part, except for a couple things. So you and I are both humans, so our DNA structures are pretty similar, right? But my eye color is brown, whereas my friend Samantha over there, hers is blue. It's the same thing in plants, and we can see that in the genome sequence here. It's coded with A, T, C's, and G's. That's not super important, but all you need to know is where this differs. It's called a genetic marker. These genetic markers indicate where the trait values differ. So these are denoted with stars down below. So the A's are the same, the T's are the same. An example here is where it's different. One example of how we would code these is if you had two C's, it would get coded with a 1. If you had a C and a T, it would get coded with a 0. And if you had two T's, it would get coded with a -1. So from these genome sequences, we build our whole data set. And so across our columns, we have these genetic markers. Across our rows, or down our rows, we have our IDs. And our whole data set is filled with 1s, 0s, and -1s. And again, we're looking to study these genetic markers because this is where it indicates the different trait values, and that's what we're hoping to study. Does everyone kind of get that idea? Okay. So what we've done so far. So my mentor, Christina Zodi, built a model called RR-BLUP. And this is showing how well RR-BLUP is doing at predicting the height value of these 6 species. The way you want to read this table, it's a heat map. And so we're using Pearson's correlation coefficient as a measure of accuracy here. All you need to know is that you want a value that's closer to 1. Instead of looking at the value, you can also just look at the color of a box. The color of the box, excuse me. So if the box is more red, that means that the model is doing better at predicting height for that species. So something like switchgrass. RR-BLUP is really good at predicting the height of switchgrass, but it's not really great at predicting the height in rice. But my mentor did not only create one model, she built 11. So there's a variety of models here. There's some linear, there's some nonlinear, there's some machine learning, but the most important thing out of this whole heat map is that there's not one model that does great at predicting the height for all 6 species. In fact, I want to point you to this last column here, which is an artificial neural network. That's a deep learning tool, and as you can see, that entire column is mostly yellow, which means that that model is not really doing great at predicting height for any of the species. So the goal for my summer was to build a convolutional neural network, which we'll talk about in 1 second. And we wanted to compare that to not only the artificial neural network, but also the other models, and we were hoping for some better results. So the next question you should have is, what is a convolutional neural network? So convolutional neural network is just a deep learning tool. It's part of machine learning. It's typically used for image classification. So we're going to talk you through this and then let you guys ask questions if you need to. So this whole structure is just a layout structure of a CNN. I'll go through the parts in a second, but I want to explain to you how it works. So if I build this model, it would be used for digit recognition, meaning it would recognize handwritten digits here. So if I pass my model this hand digit 2, it'll give me an outputted probability that it is a 2. So the outputted probability for a 0 would be something like a 0.1. The outputted probability for a 9 would be like 0.05, but the outputted probability for a 2 should be like 0.9 because it's actually a 2. The way the model does this is that it doesn't actually look at the whole 2 and say, yeah, that's a 2. It filters through different parts of the images, right? So it recognizes the loop at the curve at the top, excuse me, with a loop at the bottom, and assigns weights to where those features happen. And that all happens in this convolutional layer here. After the convolutional layer, there's a max pooling layer, and that works to decrease the size of the image, and it takes the maximum value of those weights assigned here. Typically after the max pooling layer, there's a dropout layer, and that randomly assigns some of the weights to 0, and that just helps to reduce overfitting. This fully connected layer over here just connects all the neurons that we're talking about, and the output layer gives us our probability. How are we doing? Good? Okay. But now you should say, Emily, your data isn't handwritten digits, it's the 1s, 0s, and negative 1s that you just described to me. So for our data, for our neural network, excuse me, it takes in this data and it filters through the 1s, 0s, and negative 1s, and it recognizes those patterns. So you can imagine that if you had like a 2x3 box here, it would filter through those images and recognize the patterns of the 1s, 0s, and negative 1s and associate that with a predicted trait value. This framework or this structure is an 18-16-32-1, meaning there are 8 convolutional layers, 16 convolutional layers, and 2 fully connected layers, 1 with 32 and 1 with 1 layer. This model was based off the DeepGS package, which was developed by a lab group at Northwest A&F University. We used this structure as one of our structures for our model, and we also used a simple structure, which is just one of each of these type of letters. A common thing in machine learning is that there's not one way to do it. There's not one way to solve the problem, and that kind of gets tricky. So we built both of these models to kind of compare between the two and see which one did better for what our goal was. Everyone still with me? Okay. So once we built this model, we then did a grid search. A grid search takes some parameters, and it takes, so let's say I had 4 values for learning rate, 4 for minimum delta, and 4 for patience, for example. It would run, it would take combinations of those 4 values and would run the model based on those 4 values and tell me how accurate the model is. So that's what this heat map is showing right here. Again, we're using Pearson's correlation coefficient as a measure of accuracy. You just want a model that's close to 1, or if it's easier, look for the red box. As you can see there's only 3 orange boxes, which is not really great, and the rest of them are yellow. In fact, the Pearson's correlation coefficient is actually 0.1, which is pretty terrible. Here we're only looking at things like learning rate and minimum delta, and so we decided to do another grid search with more parameters that are related to the actual structure of the model. So things like number of convolutional layers, the convolutional kernel size, learning rate, activation type, things like that. So we did another grid search with these, and we ended up with something similar. So again, we're looking for the red boxes, which is just this bottom left-hand corner. So at this point, you should be saying, Emily, it looks like your model's pretty terrible, and that's what I thought too. But it turns out, so 2 things. This is a common problem in machine learning that this just means that our model is insensitive to parameter changes. So that's not necessarily a good thing, but it's not really a bad thing either. Another problem in biology data specifically is that you typically have a lot of genetic markers compared to the number of individuals. So you can think about that as you have too many categories, excuse me. So let's say you had 100 categories, things like eye color, hair color, height, things like that, and you only had 5 individuals. So you give the model that data and you say, okay, now predict the height of the 6th person. That would be really hard for the model to do, right? Because it only has 5 individuals to base it off of. And that's kind of what we're talking about here. So we have too many genetic markers compared to the number of individuals, so it's making it really hard for the model to learn and predict different values. Does that make sense? Okay. So then we picked the best parameters based on those 2 grid searches, and we ran our model. Quick overview, so again, this is a heat map. We're running this for spruce data. This is diameter, breast height, wood density, and height. We're focusing on these 2 columns here. So the -S is the simple model, so that's one of each type of layer. The GS is the full model. If you look at those 2 columns, the model is not doing as bad as we expected, but it's really not doing great either. It's pretty comparable to the artificial neural network, especially for wood density, but it's not really doing as great for things like the linear models. So this is where my research ended for the summer, but of course research never stops. So after I left, my mentor Christina Azzotti ended up running my model for the rest of the 6 species that we were studying, and she predicted height for those values. We actually got some pretty cool results, and our CNN actually ended up performing better than our artificial neural network for both rice and switchgrass. You can see that here and here. This is actually pretty awesome because a lot of biologists first don't understand what machine learning is, and they don't really understand the point of it. So in previous to this, there's a lot of linear models that are used because there's been a lot of research done on them, and people typically understand those. And people are pretty skeptical about things that they don't know a lot about, things like machine learning. And actually this CNN model was the first one, to our knowledge, that was used to analyze biology data. So getting these results was actually pretty cool. So thank you for listening to me babble for the past 15 minutes. Again, this was NSF funded. It was a part of the ACRES REU at Michigan State University. I worked with Dr. Shin-Han and Dr. Christina Azzotti now. And if you want to learn more, you can talk to me about this, about the REU, about biology, about machine learning. And if you really want to know more, you can read the paper that was just published about a month ago. Does anyone have any questions? It doesn't have to be about this. It could be about like what is an REU? Anything? All right, cool. Thank you guys.